Oh, you're on mute.
Sorry, still I'm doing this kind of stuff. I'm sorry that, I'm from Japan and it's 11 p.m. Let me, please forgive me, that it's 11 p.m. right now in Japan. So I'm introducing today Dr. Beau Weber, who's assistant professor of pediatrics and also, of course, a member of Masonic Cancer Center and the Stem Cell Institute. And so let me introduce him briefly. So he's a graduate of University of Wisconsin-La Crosse, got BS, and after that he got his PhD from our University of Minnesota, Minneapolis, and then after doing postdoc, he's been an assistant professor of pediatrics since 2017. So he's been funded by NIH for several awards and some company projects and private foundations. And he's very well known for his work in his genome editing and its application for the T-cell modifications. So then we are looking forward to his presentation today about precision genome engineering in T-cells for cancer immunotherapy. So the podium is yours, Beau.
All right, thank you very much. Let me just pop this up. And are we good to go? We see in the actual screen here?
Yep.
That
All
looks
right. Great. All right, well, yes, thank you. Thank you for having me today. Very much looking forward to telling you about our lab's work on genome engineering in T cells for cancer immunotherapy. Do you have just a few disclosures? Some of the work towards the end of this is done in collaboration with Intima Bioscience. So our lab is interested in T cell engineering, primarily for cancer immune therapy. There's a number of areas that we work on in the lab centering around multiplex gene editing, and addressing some of the toxicity that's associated with double-strand breaks of Cas9 using base editors. We also have work going on in non-viral engineering, primarily to overcome some of the toxicity and integration efficiency and get away from viral vectors. And then we're always very interested in clinical application, and we have a current project on genome editing in tumor infiltrating lymphocytes. So for today, for the sake of time, I'm going to be talking about the multiplex editing projects with the base editor, and then ending with our work in the clinical translational space on some editing of an internal checkpoint in TIL. So I'm sure many here are very familiar with adoptive cell therapy, particularly using T cells, most notably in the form of chimeric antigen receptors, which have shown to be very effective against certain hematological malignancies, as well as T cell receptor engineered T cells. And the primary way that this is done currently and the approved products involve taking peripheral blood T cells from a cancer patient, utilizing a virus, primarily lentivirus and retrovirus, to insert a T cell receptor or a chimeric antigen receptor into the T cells. These T cells are then expanded and reinfused into the patient, often with preconditioning and cytokine support, where through the receptor, they target the cancer cells for destruction. And so there are a number of challenges still that face this therapy, despite the real promise that it has shown. One of the big problems is that it's highly personalized. So it's largely autologous, which necessitates a complex manufacturing process and makes it quite expensive. There's also the challenge of delivering the CAR or TCR, which, as I said, is typically done by virus, which are randomly integrating, which presents safety as well as efficacy concerns. And virus is very costly and complex to manufacture, long wait times for clinical grade virus, et cetera. And then finally, as yet, solid tumors do present a pretty big problem compared to blood cancers. One of the reasons for this is antigen specificity. It's difficult to find antigens expressed on solid tumors that aren't expressed elsewhere in the body. And also the microenvironment of solid tumors is highly suppressive. And so a number of approaches are being taken to overcome these problems. For instance, the development of allogeneic or off-the-shelf therapies, number of approaches to do this using alternative immune cells, or as we'll talk about in a little bit, through genome editing. Similarly, receptor delivery, getting away from the random integration and the viruses by using targeted integration and also the non-viral delivery. And then for solid tumors, so far T-cell receptors versus CARs have shown, I guess, somewhat more promise, and really stemming back to TILs or tumor infiltrating lymphocytes, which I'll talk about a little bit later, as well as checkpoint inhibitors, which have shown to be, very effective for some patients for certain solid tumors. And so our lab's very interested in how we can use genome editing to address these challenges. And so largely the field now, although there are other nuclease technologies, the field's really moved towards CRISPR-Cas9 induced genome editing. And so CRISPR-Cas9 functions as an RNA-guided nuclease. And so the way it works is that there is a guide RNA molecule with a 20 base pair spacer sequence that is complementary to a genomic target. Once this finds its genomic target, cleaves both strands of the DNA, making a DNA double-strand break. This break can be repaired either by non-homologous end joining, which is an error-prone process that's useful for knocking out gene function, typically resulting in small insertions or deletions at the target site. Alternatively, through a process called homologous recombination, a donor sequence can be co-delivered to the site where the double-strand break has occurred. And at a low frequency, for the most part, and outside of a few specialized techniques, you have recombination at this target site. And so this is useful for inserting, expression sequences, perhaps like a CAR, a TCR, or correcting a DNA sequence. But as we think about overcoming a lot of these challenges that are presented by the solid tumor microenvironment and advancing cancer immune therapies, it becomes apparent that the ability to edit multiple genes and do multiple types of edit in a cell at the same time is highly desirable. For instance, just as an example, certainly there are a number of other approaches to be looked at. But as an example, this is one approach towards making an allogeneic cell with enhanced function. So here we knock out the endogenous T-cell receptor to prevent the risk of graft versus host reaction. We take out beta-2 microglobulin. This disrupts HLA expression and essentially cloaks the incoming T cells from recognition by the host. You can take out a checkpoint inhibitor such as PD-1, although there are many interesting ones to look at, to overcome inhibition and exhaustion, and then deliver an exogenous CAR or a TCR to target that engineered cell towards a specific cancer. But the problem is when you start cutting the genome with Cas9 in multiple places, you start to compound the risk of off-target double-strand break induction, which has been shown in a number of studies. There's also a response to DNA damage, unsurprisingly, and in some cases there's genotoxicity associated with making multiple double-strand breaks within a cell. And finally, especially when you're cutting at different sites in the genome, there's the risk of translocations or chromosomal rearrangements. And this is, I think, particularly interesting. It's something that has, up until recently, not been explored very extensively. And there are a number of clinical trials ongoing with nuclease technologies where multiple cuts are being made in cells. And so one of the questions that we had was, how can we do genome editing without making these potentially problematic double-strand breaks? And one technology that really emerged to enable this was out of David Liu's lab at the Broad, and what they developed were so-called base editors. And so base editors essentially consist of a Cas9 that is rendered able to make a double-strand break, but only make a single-strand nick, so it doesn't create indels or only creates them at extremely low levels. This is fused to a deaminase. And so when the Cas9 hybridizes with a DNA sequence, you create this R loop where the two DNA strands are separated. And at this point, the deaminase is able to interact with the target strand and deaminate C's within the protospacer window. And so the initial base editors were a cytidine deaminase, so this made C to T edits. And there was some context dependency with this, but overall they worked quite well. The double strand break induction was very low, less than 1%. And then just a year later, a second paper out of David's lab, this one by Nicole Gadelli, describing adenosine base editors. And so in this case, going from an A to a G, largely working in a similar fashion, but with a engineered adenosine deaminase that they produce through protein evolution. The one benefit of ABE is that it's a little bit less context dependent, and the indel formation, the background indel formation is even lower than that of CBEs. And so we started to wonder, how can we use these base editors to knock out gene function for use in immune cell editing? And so one of the approaches that's pretty straightforward is simply converting an amino acid codon into a stop codon. There's a number of ways that this can be done, outlined here on the left. Alternatively, what we reasoned is that potentially by disrupting splice sites, you might have a more effective knockout, because stop codon read-through is well described in the literature, especially, and happens in cells, especially under stress. And so we thought that if you disrupted the level of splicing, you're kind of hitting the gene function at both like the transcriptional and translational standpoint. And so we did a number of work. I'm not going to be able to describe it here. It was actually just published by first authors are Mitch Klusner and Walker Lahr. It was accepted at Nature Communications, where we really went through and did a thorough characterization. And we did find that splice donors are very effective, more effective than inducing premature stop codons in terms of knocking out gene function. Some of this work stemmed from our initial observations in T cells, where we designed these splice donors to target the TRAC, which is the T cell receptor alpha constant locus, PD1 as a checkpoint, and then the B2M, as I mentioned, are part of the MHC. And so we wanted to use this sort of as a model system to see if we can use base editors to do multiplex editing in T cells. And so what we found through quite a bit of work, this was published a couple years ago now, was that by optimizing the codon optimization and delivery and a number of other parameters, we were able to essentially go from 40 to 50% editing at all three genes at the same time, up to a sort of our most optimal condition between 95 and 99% editing at all three targets simultaneously. And I think in the context of multiplex editing, this is really important if you start to think about the composition of the cells within a population, because there's no selection used here. You edit the cells and they are what they are. And so if you look at this is using essentially a SPICE analysis that's been repurposed for representing multiplex gene knockout, where you have sort of this middle pie area that represents the portions, I guess the pieces of the pie represent the proportion of the population that has specific edits. And then the arcs around the SPICE diagram represent the actual genes, in this case, TRAC, PD1, and B2M. And so you can see in our pulse only control, we have the majority of our pie is no knockout, obviously, as you'd expect. And then when we start to get low level editing, we can see that there's a small portion of pie that has multiplex, has T cells that have all three genes knocked out, but you get sort of this mixture of other cells where there's proportions that have, maybe one gene knocked out, two genes knocked out, et cetera. And so it's kind of a mixed population. When you start to get to very high editing rates, over 90, 95%, you can start to see where the majority of the pie becomes blank, where all three of these genes are knocked out. And so I think it's very important in this case to be able to achieve that very high rate of editing in order to make a uniform product with more predictable function. And so this base editing process, because we use modified mRNA and modified synthetic guide RNAs, is very inert on the cells. We took the multiplex base edited cells, transduced them with a CD19 CAR in this case. And what we found is that all of them essentially, no matter what what had been done to them with the base editor, had nice polyfunctional cytokine response here. This is again SPICE analysis, but now instead looking at the production of the cytokines TNF, IL-2, interferon gamma, and you can see there's a good proportion of cells that have 3 functions, which is desirable. Likewise, they were very cytotoxic. In this case, we challenged them against Raji target cell or a Raji target cell overexpressing PD-L1. And so you can see when we have no CAR, we see notice cytotoxicity on the left. When we put a CAR into these cells, you can see that although the unengineered cells are able to kill the wild type Raji cells, they aren't able to mediate a significant killing of the PD-L1 overexpressors. But when our multiplex edited cells, as would be expected, we see that they are effective at killing Raji that overexpress PD-L1, showing that this gene editing process can be used to overcome certain challenges like checkpoint genes. But one of the real key findings from this work was the fact that the double strand breaks were reduced substantially. So here, if you look on the left, we look at edited T cells that were edited with Cas9 nuclease. We can see very high rates of editing, which portend to very high rates of those indels that I had mentioned earlier. Whereas when we look at the base editors, we see that the rate of indel is extremely low, as would be expected. One interesting thing that we found is that as our editors became more efficient, we actually saw reduced double strand breaks. And so this was a really nice finding as well that portended well for downstream application. And so just thinking about this, in our case, we had, so in this case, 3 targets. We did find one very low level off target cut site for our PD1 guide. So in theory, we had 4 spots in the genome that were being cut. These, at 90+%, this one at probably like 1%, I think around 1%. So not very high, but it's there. And so you have all of these potential translocation products listed down here. And these are really only the balanced translocations. You obviously have corresponding unbalanced ones that can occur as well. But for this sake, we're looking at the balanced translocations. And what we found is if you look here in the middle row, where we used Cas9, whether it be protein or mRNA, we see that pretty much in most cases, you can detect every single combination of translocation that you can think of. The frequency is very low with the off-target site. That would be as expected because of its really low rate of indels. But some of these, for instance, B2M and TRAC, we're seeing up to 4% if you combine the 2 reciprocal species of translocation in this population, which may not seem like a lot, but if you're dosing with 1 billion, 10 billion cells, you start to get to a large number of cells that are carrying these translocations. But when we use base editor, what we found is that in this study, over a number of donors utilizing this droplet digital PCR, which has a pretty high sensitivity, we were not able to detect any translocations using base editor. And so this was a really striking finding to us and very exciting. And it was really interesting because when we look at the expansion of immune cells in culture after doing this editing, what we find is that compared to, in this case, it's normalized to a control, pulse control, so no editing at all. What we see is that when you edit with Cas9, you see a reduction in the expansion, so almost nearing a 50% reduction. We don't see that with base editors, and this is probably due to the fact that we're not cutting the genome in multiple spots, setting off a lot of alarms in these T cells. And so not only are these T cells very highly edited, uniformly edited, highly functional, they also expand very well and are just generally, I think, happier overall when we use a base editing approach. And so beyond this, as I had mentioned, a lot of the engineering strategies today use randomly integrating vectors, so lentivirus, retrovirus. And although there have been relatively few adverse sort of events from random integration, there have been some documented issues with integrating in regions that cause dropout and perhaps inhibit the function of the cell in some way. You have variation based on where the CAR integrates in the genome, so it's a little bit less predictable about how that CAR is going to be expressed. And so we were really interested in finding ways to combine this ability to knock out genes with base editor with a way to target knock-in very specifically. Now, the thing is that when we think about this, one of the approaches what we wanted to do is be able to target the knock-in and then do the multiplex editing with the base editor without making all of these double strand breaks. And so, one approach that we thought we could use is to pair the Cas9 base editor with an orthogonal nuclease. So this would be a nuclease that uses a different, essentially a different guide RNA structure. So the guide RNAs will not cross-react. And so in this case, what we have is the base editor recognizing its guides, which are designed to make the multiple edits to knock genes out, whereas the orthogonal nuclease is dedicated to making one cut at a safe harbor site where a gene can be inserted. In this case, the examples I'll show you are with recombinant adeno-associated virus. And so to sort of test this out, we use this gene trap reporter strategy that we use quite a bit in the lab. So this is essentially targeting the AAVS1 locus, which is a well-known, well-characterized safe harbor locus. And so what we do is we target a splice acceptor GFP construct here. When this integrates, and only upon proper integration, does the splice acceptor essentially co-opt transcription from this endogenous locus and produce GFP. And so what we see, in this is all in primary human T cells again, is that with the standard SP-Cas9 nuclease, we see very high rates of targeted knock-in, nearing 80%. We used another nuclease called Staph aureus Cas9. This one also was quite efficient, maybe a little bit less than Cas9. We also tried CPF1, and initially it was pretty disappointing. This was actually protein from, I think, from IDT, but then they developed a hyperactive variant of this so-called CPF1 Ultra. And in this case, this cut extremely efficiently, and it was actually, in this case, as a protein, there were some added benefits. And so, having tested these, we started to look at potential places and strategies to knock in a CAR or TCR. And around this time, maybe a little bit before, Michelle Sadelane's group published this really nice paper in Nature showing that if you target a CAR to the actual endogenous T cell receptor alpha locus, it enhances the function of the T cells. And so we thought this was a pretty slick approach and very interesting. And so we took our 3 nucleases here, tested editing at the track in T cells, and what we found is that all were very, very efficient. The CPF1 Ultra was probably actually the most efficient, nearing 100% knockout after electroporation in T cells. And so we decided to go with this strategy and test out a strategy where we use the CPF1, which is here is also called Cas12a, to do the knock-in. And so this is a mesothelin specific CAR packaged into AAV6, designed to target the track locus, and then base editor was used in this case to knock out PD1, B2M, and CISH. I guess by sort of the mechanism of the knock-in, we also knock out track. So it's essentially a 4-gene knockout with a targeted knock-in of the CAR. And what we found with this approach is that we were able to achieve very efficient knock-in at track, anywhere between mid-40s to mid-60% at the population level. And I think really importantly and interestingly is that when we started to knock out multiple genes in this setting, it did not decrease this efficiency of knock-in. The rate of knockout was also extremely high. So track was, up near the 100%. B2M knockout was really efficient, as well as PD1. And then this other checkpoint that I'm going to talk about quite a bit later in the talk, called CISH, was another one we added in that we were very interested in. And so we were essentially able to knock out all these 4 genes with very high efficiency while simultaneously knocking in a CAR. And these cells were very functional. This isn't a particularly highly challenging assay, but it's a killing assay against a target line that expresses mesothelin, and we see really nice killing even at fairly low E to T ratios. And so, this was really exciting. It worked very effectively. It's very reproducible, but it's a little bit complicated. Having to use a different Cas9 nuclease in addition to the base editor. You start thinking about scaling up, taking this over into GMP. It gets a little bit complicated when you start thinking about it at that level. And so what we wondered is, can we devise a way to use base editor as an all-in-one system to be able to do the multiplex editing and also stimulate targeted knock-in? if you recall, base editor does maintain a nickase, so a single-stranded nickase function. And so what we reasoned here, and this is based on a number of other studies with a Cas9 nickase, but what we reasoned was that the nickase function of base editor could be used, leveraged to stimulate the knock-in by essentially creating offset or staggered nicks within the genome. And so we tested this out. So again, here's our SP-Cas9 nuclease, very efficient. This is an SP-Cas9, so a non-base editor nickase. So with one guide RNA, we see this pretty substantial reduction in the efficiency of knock-in, but if we pair the two guide RNAs together, the efficiency goes up almost on par with SP-Cas9 nuclease. And so, this was really interesting and exciting. But then we started playing around with the base editors, and we started to see that a pretty substantial drop in efficiency, but still really respectable. A lot of the clinical CAR T products, you're still in the range where these cells are in terms of the efficiency or the frequency of CAR in the population. But when we actually started to use our most efficient base editor, you remember codon optimized, the efficiency went down even further. And one reason for this, we think, is probably due to some of the background indel rates that we were seeing with our less optimized editors. And so when we cleaned that up, we started to drive down the editing. And there's another reason too that I'll touch on the next slide. But suffice it to say, we saw that we had some work to do here. And around this time, it was interesting because an enhanced adenosine base editor, or ABE, was described that was very efficient in our hands. As I mentioned, ABE actually has reduced indel formation compared to CBEs, and there's a couple other benefits. So we were always very interested in the adenosine base editors, and so this one was the first one that really had like very high potency in cells. As you can see, looking at multiplex knockout of the same genes here labeled CD3, but this is the TCR complex, and then CISH knockout as well. And what we found, interestingly, is that if we take, so here we have our nickase Cas9, this is actually knocking in a CAR at ABS1. So we're up in that 70% knock-in rate, very good. Our BE4, so this is the CBE, down again in the 10% range. And what we found is that with the adenosine base editor, the enhanced ABE8E, we found that knock-in was actually improved quite a bit. And so around this time, we started thinking, if you look at where these editors edit in the genome, what's happening is these base editors sit down, they start to deaminate these target bases, and when this happens, the complex can no longer bind because the guide RNA is no longer complementary to that sequence once the actual sequence changes. And so what we came up with was this idea of utilizing retargeting guides. And so the concept here is that you design a second set of two guides whose sequence actually matches exactly the edited sequence. So after the base editor functions, these new guide RNAs are able to edit that locus. And when we tested these, we saw about a twofold increase in targeted knock-in with ABE, now up into the 40 plus, nearing 50% targeted knock-in. And so we think that this approach actually is going to be really helpful. We're making a few other modifications too. I think, with this particular strategy and a few tweaks, we're going to be able to get really close to what we see with the nickase Cas9 and get into that area where we can, really produce a pretty complex edited cellular product with a single enzyme with this method. So just to wrap up kind of this first part of the talk, what we found is that multiplex base editing is very, very efficient. It reduces double-strand breaks, translocations, and potentially some genotoxicity based on the expansion data. Targeting splice sites is most effective. And if you want to, really see a deep dive on this, I'd check out Mitch and Walker's this newest paper that just came out. It's also really interesting because it includes an algorithm, essentially a web tool that you can use for any gene, any ensemble annotated gene. You can go in, punch it in, and it will design base editor guides to be able to target splice sites to get really efficient gene knockout. We found that if you combine the base editor with an orthogonal Cas9, you can do multiplex knockout and knock in very, very efficiently. Cells are highly functional, but also now potentially use this all-in-one base editor approach, which I think is really starting to near the efficiencies you would need for a therapy. Some of the things we're working on right now, we have in vivo studies in progress to look at these multiplex knockout and knock-in T cells to really kind of stress them a little bit and see how how effective they really are in that setting. We're also interested in this concept of hyper multiplexing. Really, it's kind of looking to see how many genes can you edit with base editor in the system. We've done 4 in T cells. We've done more in other cells. I think it's going to be we'll be able to edit quite a few. And the other thing we're really interested in is moving beyond a gene knockout. So knocking out genes is great, but there's also some really interesting things you can do just by changing nucleotides or messing with splicing and causing exon skipping as well. So we've got some pretty novel ideas in that space that we're working on as well. And then finally, even though AAV is very efficient for knock-in, it's still a virus. It's still kind of costly and complex. And so we'd like to move away from that. And we've got quite a bit of work that's looking really promising with being able to do this without AAV and just using a non-viral integration method. All right, so that's the first half of the talk, and I'm going to switch gears now and talk about some really exciting work that we've done over the past few years on actually a clinical translation of a genome edited product. And in this case, the long name of it, I won't read it all the way off, but essentially utilizing CRISPR-Cas9 to disrupt an intracellular checkpoint gene called CISH in tumor-infiltrating lymphocytes. And sort of this is just the outline of going from the bench essentially to the bedside. So at the beginning of the talk, I showed you kind of the standard engineered T cell pipeline, viral transduction CAR, TCR, but actually where most of this actually began was out of the surgery branch at the NCI in Steve Rosenberg's lab, looking at tumor infiltrating lymphocytes. So TIL are essentially T cells residing within the tumor and microenvironment. They're recognizing in some form that the tumor is not supposed to be there, but as I mentioned, the tumor environment is very suppressive, and so these cells are kind of stuck in a state of not being able to clear this tumor. And so what the Rosenberg group found was that if you take these TIL out of the tumor, grow them in high IL-2 and all the nutrients that they need into very large numbers and infuse them after lymphodepletion, you're able to achieve, in some cases, pretty remarkable results even against solid tumors, particularly melanoma was one of the first that they really focused on. Alternatively, you can take these tumor fragments, cut them into small pieces, and isolate them and grow them into these kind of oligoclonal T cell populations that can then be screened for neoantigen reactivity. And this has actually been used to target other cancers, such as epithelial cancers and other solid tumors with relatively good results. And so if you look over time, this is just looking at TIL. As I mentioned, a lot of the initial work was done in melanoma with some examples more recently of complete responses in advanced metastatic colorectal, breast, cholangio, et cetera. And what you find is that you start to look at this, and what you see is that, for melanoma, you have this very high rate of mutation. And with these latter cancers, you essentially have many fewer. And so in this case, as I mentioned, that neoantigen selection can be used to enrich the TIL for reactivity against these neoantigens. And that's really critical, I think, for some of these cancers that don't have such a high mutational load. And what this really corresponds to is T cell receptors in the population that are recognizing these neoantigens. And so if a cancer has many more neoantigens, there's going to be many more opportunities for endogenous T cell receptors within the repertoire to be able to recognize those and target the cancer. And it gets, lower in probability the fewer the neoantigens you have. And so this enrichment process really helps that. But beyond that there's still a number of problems. The TIL therapy doesn't always work, especially for more of these advanced metastatic cancers like GI cancers. And really, I think, this comes down to the fact that the tumor microenvironment is very complicated. Checkpoint inhibitors have proven quite effective, but a lot of times they're limited to specific context. For instance, PD-1 blockade really is only helpful in the situation where that signaling axis is at play. And then there's other problems. So for instance, neoantigens oftentimes can be expressed at relatively low levels. They can be presented poorly on MHC by tumor cells. In many cases, again, not always, but neoantigen reactive TCRs can be of lower affinity. And then there's a number of other issues like that contribute to suppressing proliferation, persistence, the migration of T cells, et cetera. And so again, because we like to work on genome editing, we asked, how can we use gene editing to overcome some of these problems? And so through a collaboration actually with the Rosenberg group, as well as Nick Restifo and Doug Palmer, we became interested in this gene called CISH. And so CISH is a member of the SOX family. It's initially known as an inhibitor of cytokine signaling through interaction with intracellular signaling domains, and had been shown to be a negative regulator of STAT5 previously. Interestingly, though, Doug Palmer found, and Nick Restifo's group found in 2015 that CISH also plays a novel role in regulating TCR signaling through the polyubiquitination of PLC gamma. And so when the T cell is activated, CISH expression goes up. It negatively regulates the signaling process and inhibits downstream activation of the T cell. What they found is that if they use T cells from a mouse that was deficient in CISH, they saw very good clearance of, in this case, is a B16 melanoma model with the CISH knockout cells compared to wild type. And this led to a really profound increase in survival in these mice. And so the question became, does this actually take place in human T cells? And so in some of our initial studies, we took simply T cells from peripheral blood, activated them with CD3 to ligate the TCR, and then looked at CISH expression over time. And what we saw is that, in fact, yes, CISH does go up upon activation, similar to what is seen in mouse. It does seem to be linked to differentiation status, so it is activated much more quickly in effector memory cells versus naive cells. Interestingly, when you look at peripheral blood T cells versus T cells derived from a tumor or TIL, we find that although peripheral blood cells largely do not express CISH normally, TIL, there's a solid portion of them are actually producing CISH at a steady state, suggesting that it could be restricting their ability to recognize neoantigens. And so this led to a hypothesis that if we were able to knock out CISH function in T cells, we could potentially enhance their function. And we turned to, in this case, CRISPR-Cas9 for this, as drugging, an intracellular molecule is pretty challenging, and CISH plays other roles beyond its role in immune cells. And so a systemic option is not ideal. And so, we thought that this was a very good candidate for utilizing genome editing. And so we went through a pretty extensive evaluation of the CISH gene to design editing reagents, looking at just predicted specificity with computational prediction algorithms, targeting 2 shared exons. You can see that there's 2 isoforms where the initial exon is not shared between them. And so these are not ideal candidates for targeting. And so we focused on 2 shared exons. What we found, especially targeting the terminal exon, was a number of guide RNAs with very high editing efficiency at the genomic level. We ended up settling on one optimal guide RNA that was right at the beginning of the exon. And this genomic editing corresponded to very efficient knockout at the protein level. In this case, looking here at unstimulated and then stimulated T cells and the control and the knockout. And so because we were interested in translation, we were very interested in the specificity of this. And so we took 2 approaches to measure specificity. One was evaluating computationally predicted candidate off-target sites with deep sequencing, where we identified 0 off-targets. However, we also wanted to carry out an unbiased approach. And so to do this, we used GuideSeq, which is essentially uses small oligo adapters to capture double-strand breaks wherever they occur in the genome. And using this approach, we found also that no off targets were detected with this particular CISH guide RNA. Just to convince you that we actually can do this assay. This is a PD-1 guide that I had mentioned before, where we did find a single off-target effect, and we captured this both by the computational prediction and by GuideSeq. And so the assay is robust. In this case, it's just our CISH guide RNA is very, very specific, which is very good for our purposes. So the next question was, if we knock out CISH in T cells, does it do anything interesting or helpful? And so we started by taking peripheral blood T cells. We targeted AAVS1 just as an inert control, edit, or CISH in a number of donors. And we looked at interferon gamma release from CD8s at titration of anti-CD3, again, simulating binding of the TCR. In this case, we found that the CISH knockout cells produce significantly more interferon gamma across the titration, which would corresponds to what we had, was reported in the mice. The cytokine response was polyfunctional, maybe a little bit more polyfunctional, but overall remained polyfunctional with a greater magnitude of cytokine production. And when we engineered these T cells, in this case with an NY-ESO TCR, and put them against a tumor line on the top here that does not express the NY-ESO antigen, or one that does, we see that with just Cas9 alone, we see cytokine response in the presence against the line with the antigen. Pretty much a similar response in a scrambled guide RNA, but when we knock out CISH, we see this really profound increase in this case, interferon and TNF alpha producing T cells against this particular line, but importantly, not against the line that does not express the antigen. And this corresponded, really nicely with cytotoxicity in vitro as well. And so at least in peripheral blood T cells, it looked like, we were having a good response, functional response to this engineering. So the next question was, does it work in TIL? And so here, again, reiterating sort of the neoantigen selection process. In this case, tumor is taken from the patient, DNA is extracted for exome sequencing to identify these non-synonymous mutations. These are made into peptides that are presented on APCs to the fragments of the tumor or TIL grown from these fragments. And this is used to select those TIL subpopulations that are enriched with reactivity. And so in this particular case example, this was TIL derived from a metastatic colon cancer patient with reactivity to this specific mutation. We knocked out CISH in these TIL with very high efficiency. And what we found here now in this case, instead of using anti-CD3, we're actually mixing these with APCs that are pulsed with either the wild type protein peptide or the mutant. And what we see is that our CISH knockout cells have a much higher response as measured by interferon gamma production to the mutant peptide specifically, which is very important. And you can measure this also by flow cytometry. Here looking at control versus the CISH knockout, and then wild type peptide on the top, and the mutant, you see this really profound increase in cytokine production over the control T cells. And this can be quantified as well. As another example that I thought was really interesting, this particular example was a patient who had been treated at the NCI previously. And in this case, what had happened with this patient was that after the large scale expansion of the reactive TIL, the reactivity in the infusion product essentially was lost or became undetectable. And so what we wanted to see is whether or not CISH could essentially, editing could recover this loss of reactivity. And so what we did was knock out CISH in these TIL and rep them, expand them to high levels. And what we found again was that in the case of the control, here we essentially lost reactivity to the peptides, similar as what had happened in the patient. And we were able to recover measurable reactivity with CISH knockout. Now, albeit it's a low level, but I think it was still pretty remarkable that we went from essentially a product that without any detectable neoantigen reactivity with a CISH edit, we at least get back to measurable reactivity, which is a pretty striking result for us. So having went through all of this, everything was looking really promising. Our next step was to go into GMP production, and we did this over at the MCT facility in St. Paul. We utilized essentially modified rapid expansion protocol. Some of it was taken from what had been previously developed at the NCI, where we stimulate the TIL first. They are electroporated, and then they're actually mixed with either allogeneic or autologous irradiated PBMCs and grown for 14 days. And so we did a number of process validation runs. This is a set of 3 PV runs and MCT, where we had very good genetic KO, which correspond to very high protein knockout, all over 95% knockout, and still maintaining quite good fold expansion, maybe a little bit lower than the control, but I will mention in this case that the control is actually a completely unmanipulated TIL product. So oftentimes we'll do like a pulse control where the cells are electroporated, but there's no CRISPR reagents in there. In this case, we wanted to benchmark ourselves as against completely unmanipulated TIL, which is sort of the standard. And so that's what we did here. So I think, you know, compared to that bar, this is pretty impressive, still achieving a mean fold expansion of over 1,000. And so when we started to look at some of the safety measures around this, one of the things that we were really interested in was the culture long-term. So this is a 3-week culture in the presence of IL-2. Interestingly, what we find is that the CISH knockout cells do grow and persist and expand more rapidly and for a longer period of time than do their wild type counterparts. But importantly, if you take away IL-2, they completely tank and die off, which is important because we don't want, you know, immune cells that are essentially can proliferate independent of any sort of cytokine support. We also ran GuideSeq and replicated this in TIL just to be sure, and again, found no off-target editing in TIL. So from a manufacturing standpoint, everything looked really solid. And so at this point, we went to the FDA, filed the IND, which was authorized at the time. Subsequent to that, we made the decision to carry out the entire front-to-back trial at the University of Minnesota. And so this is just a diagram of the process that we put in place. So working with Dr. Emil Liu, who's the PI for the clinical trial, as well as the MCT team, and then with my lab and Brandon Moriarty's lab in UMGC. Essentially, patients are enrolled, TILs are taken to be expanded at MCT. So they're expanded as fragments at MCT. At the same time, part of this is sent for exome sequencing at UMGC. And then in our lab, the TIL lab, we screen the fragments from MCT. So MCT gives us a small aliquot for screening, and then they retain the rest of the TIL in their clean facility. We conduct the screening to identify the fragments with reactivity, and then these cells we essentially give this knowledge back to MCT. They take the fragments out that are reactive, they activate them, do the CRISPR editing, and then they go back into the clinic and are infused into the patient. And so this is just some of the data as we got this entire process up and rolling. So in this case, this is actually the fragment outgrowth this is from an ovarian cancer patient. So here's an example of 24 fragments grown at MCT. In this patient, we identified 207 non-synonymous mutations by exome sequencing. And we conducted screening of these peptides on APCs derived from monocytes. And here's just an example of reactive fragments that were identified. Here looking at an ELISpot for interferon gamma. And so you can see within these different tumor fragments or TIL populations, we identified several that had reactivity to specific peptide pools as measured both by interferon gamma and by flow cytometry for 41BB, which is an activation marker. Relayed that back to MCT. They pulled those fragments out, pooled them, did the CISH knockout, and rep them to a total cell yield of over 1 × 10⁻⁻⁻. And these cells had very good potency in this case. In fact, the KO had a little bit higher interferon gamma release, which is what you would expect. And so we enrolled the first patient in this trial in the summer of 2020. In this case, it was TIL were derived from a, this is a GI cancer patient. TIL were removed from a resected liver metastasis grown out from 9 fragments, met spec for growth. And we kind of carried out the same screening process. We identified 3 fragments with specific reactivity against the patient's mutations in the peptide pool screening. And in this case, this first patient was dosed in November of 2020. And because this is an FDA-mandated dose escalation, the initial dose is on the low side, but our editing was very efficient. This patient received about 200 million neoantigen reactive TIL with no adverse events to note. The second patient was dosed about a month later, in this case, 4 × 10⁹ TIL, so ratcheting up. Again, no serious adverse events as well. And then our third production, we were able to hit 3 × 10⁻⁻, again with 95% knockout. So in terms of the production so far, going quite well. TIL can be finicky, there'll be hiccups down the road, but things overall are looking quite well so far. And obviously it's very early days. We're too far into this. So I'll save it for Emil for the clinical report at some point, which will be very exciting to hear about. But this is just an outline of some of the correlative studies that we're doing and sort of information about the trial. So as is standard with pretty much all TIL trials, the patients receive Cy/Flu lymphodepletion, TIL infusion, and then receive intravenous IL-2 or IL-2 injections after that. So the primary outcome measures maximum tolerated dose. We want to see some preliminary efficacy. That would be great, as measured with RECIST criteria. And then, of course, any incidence of adverse events anytime with a novel checkpoint inhibitor. We're also doing some correlative studies. And so in this case, looking at TIL persistence by next generation sequencing. So this is really interesting. Previously for TIL therapies, because they're autologous, it's difficult to profile them in the blood unless you have a tetramer or some other way to maybe measure them by flow. It's not very straightforward. In this case, we actually have a molecular barcode created by the CRISPR at the CISH locus. And so we're going to be able to use that marker and deep sequencing to measure the persistence of these TIL. I think that'll be very interesting. And there's some really interesting information we can glean from that. We're obviously still working on going through all of these standard tetramer sort of staining in the peripheral blood, parsing out the reactivity, also looking at cell-free DNA and RNA, cytokine profiling, et cetera, as well as phenotyping of the TIL and a number of other correlative studies that would go along with this. And so I think with that, I'm going to end. I'll acknowledge all the collaborators, as there are very many. Brandon Moriarty, who's collaborating as a collaborator co-PI on these projects, as well as Scott McIver, who's a co-PI on the TIL work. The lab, so here in red, a lot of the people who work on the base editor and the TIL work are collaborators at the NCI. Doug Palmer, who did a lot of the initial CISH work in T cells. Dave McKenna and Darren Sumstead at MCT. And then Tim Starr, who's been really great working with us on some of our ovarian efforts, as well as the trial itself. And Emil Liu, who's leading the clinical side of this, as well as all of our funding sources. So with that, I'll end and take any questions.
Thank you, Beau. So any questions? So you can use the chat function or raise your hand to ask questions.
Let's see.
Anybody?
Let's see. I see.
Okay. Yep.
Here we go.
Yep. Go ahead.
Okay. So this is Paul Orchard. So asking, so the multiplex editing approaches are exciting. Your focus of this talk was in T cells. what is the relative efficiency in CD34 and what is the potential for scale up to achieve in that population? Very good question. Something we're very interested in. We're working on quite heavily as well. Would be great at some point to give an update on that, but the efficiency is very high. We've seen efficiencies, anywhere between 80 and 90% editing in cord blood and mobilized peripheral blood CD34s. Expansion is quite good. A number of companies, especially Beam Therapeutics, are moving forward with clinical base edited CD34 products for sickle cell disease. So far, I would say it's going to be very promising to apply the base editor technology in hematopoietic stem cells overall.
Okay, so any other question? Let me ask one question. How much, for TIR project, how much tissue do you need to start with?
It kind of depends. So it depends on where the tumor is resected from. So what we find is that if it's resected from the abdomen or the liver, typically you need more because a lot of it isn't of very high quality. There can be necrosis, etc. And so you need to try to find clean tumor specimens. If you're looking at like a lung metastasis, which are probably the most optimal source for TIL, maybe like, a centimeter cubed, I guess, pretty small. The fragments that we grow the TIL out of are probably 2 to 3 millimeters.
Still, they need a resection, right? The biopsy specimen is too small.
Yeah, so at least for our purposes and what we've experienced so far, a biopsy we think is too small. Although it's something we'd really like to. I mean, obviously, if you could do that, it would be great. But so far, it's just not quite enough material.
Yeah, there's a question from David Lagaspada in Q&A.
Yeah, so that's a good question. So just by NGS, no, we can't. We can really sensitively detect if they're persisting, and we can actually sort. So there's been some work out of the Rosenberg lab as well as others that show if you look at, for instance, expression of PD-1, 4-1BB, and other activation markers on circulating T cells in the peripheral blood of patients with cancer, they are enriched in neoantigen reactive TIL. So one of the things we're interested in is sorting subpopulations of T cells from the blood, and then doing the deep sequencing on those. But to your question, if you really want to basically link a specific reactivity out of the selection to the reactivity, you need to basically parse out, make a tetramer, and confirm that that specific T cell with this T cell receptor that's reactive to a peptide is persisting. So I think it's a surrogate. And it's really informative, but you need to use a tetramer after parsing out the peptides. Yeah,
there are two people raising their hand. One is Jaime Modiano. So Sandra, can you turn on the mic for Jaime? And the next will be George Aslandy.
Okay, I'm just trying to get to, there we go.
Jaime's on, yep.
He's ready.
Thanks, guys. Can you hear me?
Yes.
Yep, we can.
Great, thank you. Yeah, hard to type on an iPad. Beau, that was really exciting, really fun. And it's cool to see how many things you can edit at once. And I think that the amount of work that you're doing by trying to sort out the heterogeneity by looking at these fragments is really phenomenal too. And I think, that gets me back to thinking to the immunosuppressive environment in the tumors, and that even as you might be knocking out PD-1 and the T cells or CISH, that, all the other things that are repressing the immune response and those T cells that actually manage to get into the tumor are still present, right? So are you guys thinking about trying to combine the TIL approach with something that remodels the environment and makes it more akin to responses like depletion of immunosuppressive macrophages or alterations in the rest of the myeloid compartment, or potentially blood vessel normalization, any of those other things that might render your now more active TILs capable of really working in that tremendously immunosuppressive environment that they may not be able to overcome by simple knockout of one checkpoint.
Yeah, no, I totally agree. I mean, it's something that we're very interested, as I kind of mentioned. I mean, that's one of the reasons why we want to be able to do this really, complex multiplex editing, to be able to overcome multiple things, because that's really what it's going to take. As far as like remodeling the environment itself, we haven't dove into that a whole lot yet. I mean, it's something we're very interested in. We have projects where we're actually working to do gene editing in monocytes and other primary cells. And we do have some interest in potentially combinatorial like cell products that perhaps could get at that. I think that there's some in the field who would say that if you can get around sort of that initial inability of T cells to respond and activate to an antigen, if you can overcome that, get the T cells to really light up and start producing cytokines, that in and of itself is going to play a role in changing the microenvironment. I don't think it's probably going to be enough, necessarily, but it's something we're very interested in, and certainly would, like to explore that avenue eventually.
Okay, so then George just randomly rose his hand.
We have one in the chat.
Yeah.
Okay. Yeah.
Yes,
I saw
David
David asked one other question about the P53. It's a pretty quick one. We've seen other groups and other data that if you induce Cas9, you do see P53 induced response, but I don't know if that's necessarily why they're not expanding. We haven't like confirmed that mechanistically. It's probably a good possibility.
Yeah, and there's a chat.
Okay, yeah, here we go.
Video. Yeah.
Oh, sure. So the question is, how long does it take to find the neoantigen-directed T cell clones and then to scale up? So right now it takes about 6 weeks to expand the fragments to enough numbers to do a sizable rep. We essentially do the exome sequencing, synthesize the peptides, and conduct. So far, we've been able to do those in the same time frame. So basically, to the point where we have identified neoantigen fragments is about 6 weeks. And then it's roughly a 2 or so week process to rep the cells. So it is longer than your traditional CAR T product, and it's something that we're trying all the time to make more efficient, because these patients are very late stage metastatic cancer, and time is extremely important. So yeah, I'd say 8, 9, 10 weeks depending on how things go.
Okay, the next is the last question from George. Can you speak up?
Yeah, Beau, can you hear me?
Yes, I can.
Great talk. Thank you very much for presenting this data. I have a question. What reaction on your comments that you guys like to switch to non-viral delivery? And so as you know, I work with AAV vectors, and we use them efficiently for many cells, including T cells and hematopoietic stem cells. But did you guys try to figure it out why you need such a high MOIs for T cells targeting? Because what part of the is it intake, trafficking, gene expression? Because if you solve this problem, that you significantly can reduce cost of your production. And I don't think it will be more effective if you switch to non-viral vectors.
Yeah, I mean, AAV is fantastic. I mean, it works great. But you're right, the MOI is extremely high. I don't know why that is. I would like to know. I mean, if there, we've tested out some, kind of tricks to try to get away with using a lower MOI, but nothing like really, mechanistic or changing the AAV vector or anything itself. In our hands, we still just use a relatively high MOI to get the job done. I think one of the things for why we're interested in the non-viral is really the cost and time it takes to produce the GMP scale virus. Like for lentivirus right now, if you want to go all the way from like testing to like a GMP batch down the road, I mean, you're talking, especially through a CRO, at least a year, if not longer. If you're a company, 18 months probably. And, I've heard numbers, approaching $1 million for a batch, to do a trial. And then if your titer isn't great, you might only be able to treat fewer patients than you hope. So I think that's why, as opposed to a plasmid, which is much less expensive and only takes a few months to produce GMP scale. So I think that's one of the reasons why non-viral is really attractive. Of course, if you can take AAV and make it much more efficient and much less expensive, then I'm all on board with it because it works great.
Probably something we can work together on.
That'd be great. I'd love to.
Thank you.
Yep. Thanks.
Okay, so time has run out. So thank you very much, Beau, for presenting such exciting work. So thank you very much, everyone, to attend. And this is let's conclude this the seminar today. Thank you very much. Have a nice day.
Thanks, everybody.